Hi everyone. Let's see how many people are coming on. I'll give it 5 minutes to make sure that everybody is signed on to the quiz. The link is in the chat. Jacob has just popped that in for you. So if you click on that link, it should take you straight through to the quiz. Or if you have KwizzBit open, you can just type in the pin that's on the screen there. Whatever's easier. How are you guys doing? Exams have started now, haven't they? Was it? Yeah. I don't know if it's A-levels or AS, but Lucy's back. Have you got your flashcards, Lucy? I don't know which ones you went for. That's good. Ah. When's the first biology AQA? Is it the 9th of June? It's coming up soon. How's revision going for everyone? Oh, OCR is the 9th of June as well. It'll be good. Once you start ticking off exams and getting them out of the way, it feels good. You don't necessarily have to have the live stream visible, no. But it's handy. It's there if you want to ask any questions. We'll give it 3 more minutes and then we'll start. We've upped the subscription on KwizzBit this time so that everyone can get on, although it looks like we might not need it unless people tune in late. So if you hadn't seen the description for this quiz, we'll be going over AQA paper 2 and paper 1 revision, but it is by no means a comprehensive revision of every single topic. It should just be like a break from your normal revision, but something that's still kind of relevant to what you need to cover. Hello. Please chat away in the chat box, by the way. It's nice to see what you guys are saying, what you're up to. A couple more minutes. Hi, Ewan. Welcome back. I see we've got Stephen Grant again. I'm trying to see if I can recognize any of the names from last time. There will be some general knowledge, but I have toned it down a bit. You might be pleased to hear, you and there'll be 2 general knowledge rounds and 4 biology rounds. So it'll be 2 biology rounds, a general knowledge, another 2 biology, and then we'll finish on a general knowledge. So for today, we are going over all of the topics that could come up in your AQA A-level biology paper 1 and 2. But yeah, like I said, it's not comprehensive, so please don't use this as your only form of revision for paper 1 and 2. How do you join? Luca, if you scroll back up through the chat, there's a link. If you click on that link, it should take you straight through to the quiz page. I think a lot of this will apply to OCR too. There are a lot of similarities, lots of overlap between the 2 exam boards. So if you're doing OCR, then this should still be just as useful. Okay. So it's 10 questions per round. Yeah, finish it around 7. I don't think it'll take too long because the questions themselves don't take that long. I couldn't amend the timer, by the way. I know last time we had, was it 10 seconds or 15 or 20 seconds to answer questions? I couldn't figure out how to change that. So unfortunately, that is the same as it was last week. But hopefully some of these questions will be kind of challenging, maybe. Okie dokie. We'll start the quiz because it's gone 5 past now. Okay, everybody ready? Okay, I'm going to share my screen this time so you guys can see what's going on. So the first round that we're going to look at is biological molecules. And these are all evil questions, so if you get them wrong, you will lose points. So really think about it. Okie dokie. Oh, sorry, I need to click that. 20 seconds. Let's go. So maltose is a disaccharide made up of which 2 sugars? So the answering will be after I click through. Okay, 39 people answered. It's glucose and glucose. Fun fact, maltose is created in seeds and other parts of plants so that as they can break down stored energy for the plant to grow. Fun fact. Okay, next question. What's the term for a carbohydrate that's not oxidized by a weak oxidizing agent? If you have any questions, by the way, about the questions, just let us know. These shouldn't be too challenging. I think this is just a good way to refresh sort of the easier things. Non-reducing, well done. Lucy's back at the top. Okay, next question. Triglycerides don't affect water potential because they are which of the 4? Remember, the quicker that you answer the question, the more points you get. So don't wait until the 5-second countdown at the end. Okie dokie. Also, there is a question style later on that asks you to put things in order. I've not done one of those ones and I'm not sure how it's going to turn out. So I might have given you the answer in the question, but we'll figure it out. Okay, so triglycerides are non-polar, so they don't affect water potential because they cannot dissolve. Next question. Cellulose chains align using what to form what? So select 2. Make sure you put them in order, if that makes sense. Let's have a look. It's hydrogen bonds and they form microfibrils. That's one I always just get confused on with macro and microfibrils. Just make sure that you look out for that in the exam. Okay, how many amino acids occur in proteins that bind to the central carbon of a peptide, which is indicated in this image by R? This is an R group, by the way. A little bit of overlap with chemistry here. Answer for that one is 20. There are 20 amino acids that could make up this R group. Okay, Damla at the top for that question. Lucy Lou at the top for overall, and Stephen. Right, next question. This symbol here just means quaternary. So the quaternary structure of proteins forms when multiple polypeptide chains join together. What's the name of the non-protein molecules that bind to these peptide chains? Oh, Matt. See, I think the good thing about doing easier questions in quizzes like this is it shows you some of the stuff that, like, you might accidentally lose marks on things like this in the exam. It's like all the stuff that you need to have absolutely locked down. These are, of course, prosthetic groups. Right, how many have we got left? A few more left in this round. So when DNA helicase unravels the DNA double helix, new strands of DNA form on the exposed strands. In which direction? I did write some of these questions with kind of like a bit of an examiner's hat on. Some of them are to kind of catch you out. The prosthetic group, for the last question, that's just a non-protein molecule that has that binds to a protein, to protein chains. Okay, the answer for this one, it is opposite directions. Next question. Okie dokie. A sample of DNA is labelled with radioactive nitrogen and allowed to replicate 3 times using radioactive nucleotides to synthesize new strands. What would the percentage of radioactive DNA be after 3 replications to the nearest whole? I have a feeling this one might not have updated. So if you get this one wrong, that's on me, because I changed the answer to this. Okay, ignore that one. Oh no, sorry, yes, this one's fine. I think. Has that confused anyone? That's confused me. So after 3 replications, 8 strands of DNA will have been produced for each original strand. And 7 of these would have been, no. Okay, I'll go back through that one. We're going to be doing a revision session, by the way. This is an awkward time to say it, I'll say again at the end, but we'll do a paper 2 revision session like this one. So I'll be going over the topics that have been given in your advanced exam information, like the hardest parts of those, and sort of just going through and giving tips on revising those. So we can have a look at this then. Don't worry, honestly, don't worry about that one. Sorry, I retyped it and it obviously didn't save. Okay, how many hydrogen bonds form between A and T in DNA? Don't worry if you didn't understand that last one. I changed it earlier, but it did not update. Okie dokie. 2 hydrogen bonds. So these A and T are a base pair, and the shapes of them are complementary so that they can form 2 hydrogen bonds to make a pair. Okay, pH, temperature, and enzyme concentration should be what in an experiment looking at how substrate concentration affects the rate of reaction? Some practical tests here. That's another thing. Make sure, if you haven't already, that you take a good look at your practical handbook and that you know all of your practicals that you will be tested on, why you do them, which parts of the practical need to be controlled, and so on. Practical knowledge is where marks often get dropped. They need to be controlled. Well done, Ewan. You're at the top for that one. Okay, we're going on to cells now, round 2. So 10 questions covering the cells part of the spec. Which statement about prokaryotic cells is false? Have a read through them all. Consider them all. I'm sure your teachers have told you this, but when you have multiple choice in the exam, make sure that you go through each one and weigh up all of the options before you put pen to paper. Because even though the top one might be the correct one, you might miss out one further down, like you might autofill and think that the first one you read is correct. So make sure that you look at all of them. So the false statement was the peptidoglycan cell membrane. It's the peptidoglycan cell wall, not cell membrane. Pili are like the little tails that come off them. Okay, which organelle does the following? 1, hydrolyze objects ingested by cells, release hydrolytic enzymes to break down extracellular debris, and break down old or damaged organelles. Which organelle does all of that? So villi are structures in the intestine. So they're the things that kind of stick up. Pili are what you would find in a prokaryotic cell. Hey Zainab, not at all. Jaco can post the invite link for you for the quiz, see if you can join in. Does that make sense, Ewan? Yeah, okay. The answer for that was lysosome. That is a membrane-bound organelle that contains digestive enzymes. Okay, what is distinctive about the nuclear membrane compared to the eukaryotic cell membrane? Thanks, Jaco. There you go, Zainab. Some revision of the basics again. It's a double membrane. So the nuclear membrane is double. The eukaryotic cell membrane is only a single membrane layer. Okay. What's the resolution of a light microscope? So remember, resolution is the ability to distinguish detail. So it's the minimum distance at which 2 points of a specimen can still be seen as separate entities. It's the same as the wavelength of light. Oh, that's all right, Beatrix. It senses itself, I'm afraid. It's 0.2 micrometers. Well done, Ace, at the top there. And Caroline, I'm assuming the X's are O's. Caroline Forbes at the top. Okay, which statement is false? Again, make sure you read through all of these, consider all of the options, have a good think, and also try and answer as quick as you can. Hello, Mr. AB. So the false statement there was that viruses replicate by binary fission. They do not. Viruses replicate using the host cell to produce new copies of themselves. Remember, viruses aren't living themselves, so they don't replicate by binary fission. Okay, what do we call indirect active transport? Another very simple question, but it's a good one to make sure you have all of these differences clarified. This quiz should be helpful for you if you're doing OCR. There are lots of overlaps, but don't panic if you see a question and think, I've never seen this before in my life. We started at 6, Beatrix. Okay, the answer for that one was co-transport. So that's the coupled movement of substances across a membrane via a carrier protein. So co-transport is indirect active transport. What do T killer cells release to kill pathogens? Oh, yeah. I think if you've not seen these before, Ewan, you might want to step up your revision game before the exam. Okay, so the answer for that one was perforin. So that's a glycoprotein that produces pores in the target membrane to help break down the pathogen. Really important in the immune response. Okay, which part of the respiratory system is home to ciliated epithelial cells? Indirect active transport, yeah. I'm not sure if immunity is on the advanced exam information, but remember that anything that comes up in the exam, advanced exam information isn't like all that's going to be in the paper. Okay, the answer for that one was the trachea. And there are 6 rounds in total. We're on round 2. Okay, cysteine are membranes that provide a large surface area for which organelle? Or which of these organelles? Okay, the answer for that one is the endoplasmic reticulum. So this is kind of just tapping into your overall general knowledge of the different organelles within the cell. It kind of helps to know this kind of thing, because if you know that, if you know what the cysteine are, and you're asked about a diagram that looks at the endoplasmic reticulum, like you just know what you're looking at straight away. So it might not be something that's necessarily in your spec or something that loads of questions are going to come up on, but it just helps you a lot when it comes to those kinds of questions. Okay, during mitosis, when would you observe 2 distinct nuclei within the same cell? Yes, the mitochondria have cristae, not cysteine. It's kind of a trick question, something to look out for in the exam. With biology, there are lots of similar terms, so make sure that you know the difference between them. And the answer for this one is telophase. That's just before the cells split, you would observe 2 distinct nuclei within the same cell. Okay, time for a general knowledge round. Ewan, I'm sure you're looking forward to this one. Okay, who is Norma Jean Mortensen? Which of these ladies? T for 2 nuclei, that's a good way to remember it, Beatrix. I like that. Okay, the answer for that one is Marilyn Monroe. This is just a way of turning your brain off for a second before doing more biology. Okay, what was the most streamed Netflix series of 2021? Did you get that one wrong? That's all right. If you get non-biology questions wrong, it's not too bad. Okay, answer was, oh, the answer was Squid Games. Okay, Blue Ribbon Sports was the original name of which sportswear brand? I didn't know this before I did this question, so I'll be surprised if anybody actually knows the answer to this. I'll be very impressed, because yeah, that question did scam you. I'm sorry. You've heard it before. Are you ready to find out? It's Nike. Nike? How do you guys say it? Nike? Nike or Nike? It does look like Reebok. That's what I thought. That's why I put Reebok in the question. I was like, that's gonna trick some people. Okay, what year was Valentine's Day first celebrated? The hint for this one is that Henry VIII was the king at the time. Yeah, Nike like bike. I think I say Nike. I don't know if I use them like interchangeably. Depends who I'm talking to. Okay, the answer for that one was 1537. Well done, Ewan. You got that one. Okay, Lella Lombardi was the first female what to score points in her sport? She's a very cool lady. Does anybody know the answer to this one? Reveal answer. Grand Prix driver. There haven't been many female Grand Prix drivers. Anybody like watching Formula One? I've got a bit obsessed with it this year. Next question. Okay, how long does an elephant pregnancy last? It's the longest of any living animal, I believe. But which one is it? What are you guys going to go for? It was 22 months. Isn't that mad? Is Lucy going to win again? You guys need to catch up with Lucy. She's already got one set of flashcards. Okay, this is a zoomed in quiz. What's this? It's a picture of something zoomed in. Remember, you get more points if you answer quickly, so I think there's 4 of these questions overall, so the next 3, see if you can go on instinct answer as fast as you can. A banana. Funny looking bananas you're eating, Jaco. Oh, what is going on? This is a carrot. Sorry, guy, I don't know what's going on here. Scanned again. I'm sorry. At least this is happening in the general knowledge round. What about this one? Permission fruit? Do you mean passion fruit? I really hope this one has points on it. I really hope it does. Broccoli. Okay, thank God that one has. Okay, next question. What is this? So it's really hard to find zoomed in pictures of stuff and then also think about all the different options of what it could be alongside the correct ones. That was cereal. Okay, last one. This one I think is the hardest one. Oh, a persimmon. That's a good guess, persimmon. Okay. That was cork. That was the trickiest one, I think. Okay, let's go back to some biology. This is organisms exchange substance with their environment. Okay, which feature of the guard cells is central to their ability to control water loss from leaves? So 2 guard cells make up the stomata or a stomata. So the answer for that is the cell wall. So remember that the inner wall is thicker than the outer wall, which is why they're crescent shaped and allow the opening to open and close to allow water loss or prevent water loss. Digestive enzymes are used to extract nutrients from food. Protease breaks down protein. Which pH is optimal for this enzyme? The vacuole is as well, but it's just that difference in the cell wall thickness of the guard cells that kind of is really what helps them do the job that they do. Answer for that is pH 1 to 2. So that's the same as the hydrochloric acid that's found in the stomach. And remember that your stomach has got a mucus lining to prevent you from digesting yourself. It's good to know. How many oxygen molecules can hemoglobin bind to? 1, 2, 3, or 4? How are you guys finding this, by the way? Is it too easy? Too hard? Just right? That's 4 molecules. Okay, next question. You finding it good? Okay. I don't want it to be like too hard because then it's kind of, yeah, a struggle. But I don't want it to be too easy as well. Okay, which of these would be an adaptation of an active animal? HB stands for hemoglobin, by the way. It was quicker to type HB than hemoglobin. It's carbon monoxide, I think, that reduces oxygen binding in hemoglobin. If I remember correctly from my A-level biology days. Yes, carbon monoxide. Okay, so the answer to that one is hemoglobin would unload quicker, so it unloads oxygen quicker, which is helpful for rapid respiration to provide the energy to contracting muscles for an active animal. So something like a cheetah, if it's running really fast, it needs its hemoglobin to unload that oxygen so it can use the oxygen for muscle contraction. Okay, why aren't petometers fully accurate? And this is select 2, I believe. Again, this is another practical question, something to think about in terms of practical questions. A lot of the time, they do like to kind of see your reasoning and understanding of practical concepts so that you haven't just sat in the lesson and watched while the practical's going on, you've actually absorbed some of the info. So you get a loss of water in photosynthesis and a loss of water in metabolism. So that's why petometers aren't fully accurate. Did I put split? It was meant to say water spilt. I've not done a very good job of this one, have I? Okay, what type of epithelial cell makes up the wall of the alveoli? A petometer is like that. So it's like a tube that you have dipped into some water and it has an oxygen bubble in it, and basically you put a plant at the end of the other end of the tube, and the transpiration as the plant takes up water can be measured by how far along the tube the air bubble moves. Okay, let's reveal the answers. That's flat squamous. Next question. So there are 4 gaps. I think this is the one where you have to put them in the right order. But I'm not sure, I can't see from this end. If you're struggling with some of these words, so this live stream gets recorded. Afterwards, have a look back through and just note down all the words that you're struggling with, and then have a look at where these are on the spec and just make sure that you get to know them. Okay. So the answer for that one is the gallbladder stores bile, which is secreted into the small intestine to help digest lipids. Next question. Okay, have a look at this image. Which blood vessel is the hepatic artery? I wish I could help you with physics. I didn't do physics myself, so I'm not the best person to help out with that. But we do have physics revision live streams. If you keep an eye on our YouTube channel, we usually have weekly live streams that kind of cycle between biology, chemistry, and physics. Have a look at those for physics help, because then you can ask questions as well. So the answer for that one is D. So the hepatic artery provides oxygen and nutrition to the liver. Okay. Don't worry if you aren't getting, well, I can see lots of you saying that you're doing AQA and this isn't making a lot of sense to you. Okay, we'll carry on. Don't worry if this isn't stuff you've gone over. I did use the AQA spec as well as some of the SnapRevise questions, but yeah, we'll review. Okay, during diastole, the semilunar valves are what? So remember, diastole is the phase of the heartbeat when the muscle relaxes and allows the chambers to fill up with blood. So what are the semilunar valves doing when all that's going on? They are closed. Brilliant. Remember, that prevents the backflow of blood. Okay. Which of these substances is not found in tissue fluid? We've only got one more biology round after this, and then it's one general knowledge, and then we're done. Okay, the answer to that one is plasma proteins, because they are simply too large to fit into, well, to fit through the capillary walls. Okay, this is the last biology round. So genetic information, variation, and relationships between organisms. So what does non-overlapping mean when we describe DNA? Non-overlapping simply means each DNA base is only read once. So the same letter isn't used for two different codons. The triplets are distinct from each other. So like part of one triplet won't be read with part of another to make a different amino acid. It's just each DNA base is read only once. Okay. What term describes the duplication of one chromosome when chromosomes don't separate during meiosis? So this is what causes Down syndrome and other chromosomal abnormalities. So we're looking for the term that we use to describe when this happens. Okay, non-disjunction was the answer. Good job, Beatrix. Okay, bird migration is an example of a what adaptation? So this is an example of a behavioural, oh my goodness, I spelt that wrong as well. I'm so embarrassed, sorry guys. But anyway, it's a behavioural adaptation. So this is an innate behaviour. We don't know the exact reason why, but birds move to places that are suitable for their requirements at that time of year, and that's a behaviour that they exhibit, and it's something that has evolved. So it's a behavioural adaptation. Right, next question. Chromatin is made up of what and what? Again, select two. There are two correct answers in the four. Two, one. And that is DNA and histones. Right, who's in the top 10 overall? Caroline Forbes, brilliant. And then Carrier, then Sophie Wilkinson. Good stuff, guys. Okay. What type of mutation is shown here? So the original sequence is this one, and it's mutated to produce this one. So what's going on? Ignore the size difference, by the way. When I made the image, I made it bigger, thinking I could just shrink it so it matches, and it didn't quite work out that way for me. That's a deletion. So you'll notice that one T has been cut out. Next question, which of the following statements about meiosis is incorrect? Do you think grade boundaries will be low this year? I think well, according to like the government advice, the grade boundaries will be lower than pre-pandemic levels, but not quite as low as 2020. I don't think that the advanced information itself will have made a difference to grade boundaries, but like, I don't know if you guys know how the grade boundaries are set, but they're decided after the exam has been taken and after papers have been marked, because basically what they do is have a look at all of the marks that everybody's got, and then they stratify the marks by kind of what they expect people to be performing at. It's really complicated. So yeah, things with exam grade boundaries, you can't really predict until we know what they are. This is the incorrect statement. So meiosis is involved in asexual reproduction, not sexual. Okay, antibiotic resistance is a result of what kind of selection? Have a look through all of the options and then decide. It's directional selection. So it's an extreme trait that bacteria exhibit, and that's obviously favoured in an environment where antibiotics are being used to kill off the bacteria. Any that are left that have resistance are an extreme value of that trait. Okay, this is one that I'm not sure how it's going to work, but we'll see. Place these in order of magnitude. So this is kind of a bit of taxonomy, looking at species. You know, this is the one thing that I remember from my A-levels. I'll tell you how I remember it in a minute when we look at the answers. So they go domain, class, family, species. This obviously isn't all of the options that you could have in the exam. So it goes domain, kingdom, phylum, class, order, family, genus, species. Oh, Ewan's put a good one on there. I used to do it as Dem King prawns can't order fuzzy green sausages. Oh my gosh, I'm loving these ones. Dear King Philip came over for good spaghetti. That's a good one. King prawn curry or fat greasy sausages. Whatever works for you is the best one. Okay, What word describes the number of different species that are present in an area at a given time? Which of these is the correct word? I would ask what the rude one is, but depends how rude it was. Nice, Carmen. Okay, that is species richness. Right, last question in this round. Which of these is not a purpose of phylogenetic trees? Oh, if it's got a swear word, then don't share. I'm sure I could probably guess. So which of these isn't a purpose of phylogenetic trees? Remember to read through each one, have a look at each one. So the answer is to show similarity in species phenotypes. Remember, it's a phylogenetic tree, so we're looking at the genetic relationships between all of the species that have been plotted on that tree. Okay, this is the deciding general knowledge round. How many English monarchs have been called Edward? And this distinguished gentleman is one of the Edwards. You did well on the history one last time, Ewan. It's 9. 9 Edwards. Good job, Beatrix. Okay, what is a Manx cat missing? Yes, the Horrible History song. I used to, when I was at school, I used to print off the Horrible Histories lyrics and then like practice them at home. It's still a classic. I still watch it on Netflix from time to time. Such a good series. Okay. The Manx cat is missing a tail. Next question. Dendrophobia is an irrational fear of what? Dendrophobia. Anybody out there got dendrophobia? Actually, I'd imagine this is quite a crippling fear to have. Dandruff. No, that's a fear of trees. Didn't know that that was something people were afeared of. Okay, where would you find the Ross Sea? Oh, Matt, you've got the opposite of dendrophobia. What, that'd be dendrophilia. Where in the world would you find the Ross Sea? Anyone been to the Ross Sea? I haven't. I'd like to go. This is in Antarctica. Okay, in Gavin and Stacey, how many times had Stacey been engaged before she was engaged to Gavin? Love this show. Did you guys watch the Christmas special a few years ago? I enjoyed it. Although since James Corden went to America, it's just, I don't think it's quite the same. Never watched Gavin and Stacey. It was 5 times. That was a safe bet, Ewan. You were right. Okay, which planet is closest to the sun out of these? You live under a rock. I love general knowledge. I think it really does highlight how little you know. Mercury. There was a trick question in there. Pluto isn't still a planet. Lost that status. Quite a while. How long ago was that now? When was Pluto no longer a planet? I remember I was devastated. I don't know why. It has nothing to do with me or my life, but okay, what is the longest river in the world? Was it 2016? I should remember because it really did have an effect on me. Maybe we should start a petition for it to become a planet again. I'm sure loads of people did that at the time. Okay, the longest river in the world is the Nile. How old do you need to be to stand for US president? Yeah, absolutely, justice for Pluto. Oh, 2006. That's a really long time ago. Okay, the answer for this one is 35. You have to be 35 and a US citizen to become president. Okay, what year was Instagram launched? Speaking of Instagram, we are on Instagram. I don't know if you guys follow us yet. We post sort of test yourself content, but you might find handy a little bit of revision as you scroll through your feeds. 2010 was Instagram. Okay, I don't know if there are how many more questions we have. Which city is Banksy strongly associated with? Yeah, Facebook was way before Instagram. 2004. I watched the Social Network a couple of weeks ago now. Never seen that before. It's a good film if you haven't seen it. The answer to that is Bristol. Anybody from Bristol doing the quiz? Okay, we're finished. So we don't have Lucy at the top this time. Somebody else has taken the crown. Henzin. Is Henzin in the chat? If so, let us know. What Welsh Member of Parliament who resigned because he's Banksy? When was this? Yes, Henry. Amazing. You win. Yeah, Caroline was winning for so long. It's savage, is KwizzBit. We take it down to the very final round to decide who wins. Okay, Henry, if you want to send us an email, Jaco will put the email in the chat for you, and we will get you your flashcards. Just let us know which subject and which exam board you want them for. There you go. hello@snaprevise.co.uk. That's for you, Henry. Amazing stuff. Don't forget, guys, that we have got the seminar recording, so the revision seminars we did a couple of months ago now. You can purchase the recording of those to use in your own revision. And you also get all of the resources that people that actually attended the seminar got. So you get a booklet and the ability to ask questions about any part of your spec that you want some extra help with. More info about the flashcards there. Thank you, Jaco. And also, I mentioned earlier on that we're going to be doing an AQA paper 2 revision session, which will be ready for your exam on the 17th of June. So we'll do the live stream on the 15th of June, which is a Wednesday again. if you could let us know, so on the community tab on YouTube, you can comment on this live event, I'm pretty sure. If not, we can put a post on there, but let us know what sort of stuff you want to cover or you want us to go over in that revision session. So at the moment, the plan is to have a look at what is on the advanced exam information and kind of picking out the trickiest topics on that and going through ways that you can get exam ready on these topics. So yeah, just let us know in the chat or over on our social media channels. We are at SnapRevise on Instagram and at SnapRevise on TikTok too. And yeah, hopefully we'll see you guys there again. There won't be a live stream next week because I'm going to be away, but the week after that will be the paper 2 revision session. Thank you all for, we won't be doing one for paper 1 because I'm actually going to be away, unfortunately, for next Wednesday, and that is sort of the next opportunity to do a paper 1 revision session. But yeah, the paper 2 we will go over, and then paper 3 as well after that, but we'll discuss that in the paper 2 session. Lovely. Thank you guys so much. We've managed to do it in an hour. Hopefully that was a nice break from your revision. Keep an eye out for more of these. You guys seem to enjoy them, so, and it's nice to hear from you all as well. Thanks. Good luck if you have any exams this week or before I speak to you again on the 15th of June. And ask questions on YouTube videos. Remember, we've got tons of videos that help you with revision in general. Just look for any that have a thumbnail with my face on them. With this t-shirt on, actually, we just updated all of the thumbnails. So if you see me with a pink t-shirt on any of the video thumbnails, have a click and have a look at the advice that we give in those. And comment if you have any feedback or any other questions. And I will get back to you as soon as I can. Now you're ready and pumped to go study. Good. I feel like this is a nice soft start to a revision session. Have a bit of fun and then focus on the bits that you need to do. Remember, you can always watch this live stream back again. So if there were any bits that you were like, what on, then you can just go back and have a look. That question about the DNA strands can be completely ignored because I actually made it more confusing by trying to change it. Yeah, have a nice chill out, Ewan, before your chemistry tutor comes along. You should be able to watch the videos. So any videos with my face on them are generally sort of broad advice for revision. So you can check out those. And you can also dip into some of the biology playlists, have a look and see. I don't know, I can't answer your question precisely because I'm not sure whether there would be a crossover between what you're doing and the stuff that we have, but definitely just kind of, yeah, give them a go. 3 tips for 2 weeks before the exam. I do have a video, a cram revision video. Basically, my best tip will be looking after yourself, so making sure you're getting enough sleep, eating well, drinking well, have everything in order, so know exactly when your exam is going to be and all the equipment you're going to need for it. And then in terms of cram revising, go through your spec, write down all the topics that completely terrify you and organise them by whether they come up on the advanced exam information and then how desperately you need to cover those before the exam, and just work your way through. Remember, you will never feel ready for the exam, and trust me, that's after doing A-levels and university exams, you will never feel ready. So just do as much as you can without completely grinding yourself into the ground before, and you'll be fine. Exams aren't that bad, honestly. And if they are bad, you can always take them again. It's never the end of the world. Doesn't feel like it when you're doing A-levels, but you don't need to get it right first time. I don't know about getting grades overall. I mean, if you get 100% in one paper and then a grade B in another, then maybe you can you can put your grade up a bit, but I'm not sure. One more question. Okay, one more question and then I'll end the stream. Remember, you can ask questions on the community tab or under videos. I still struggle with exam practice. Content is fine for me. So Kitsono, we have a video about exam practice. Have a look on our channel for the exam practice video. Basically just doing past papers, but also having a look at the mark scheme and try to have a look at the way that the marks are given. So you will notice, if you go through loads of different mark schemes, you'll notice there is a pattern to the way that questions are marked, regardless of the content of the question. So yeah, have a look at that video. And yeah, keep asking questions on any of the videos if you have further help, further questions. Perfect. Thank you, Jaco. So that's a whole playlist of videos you can work through, and hopefully that helps. Thank you. I hope you have a great, oh, thank you so much. Thank you. I hope you guys have a great rest of your day and a great weekend too. I'll see you when I'm back after next week. So I'll see you on the 15th of June. Thank you all so much, and congrats to our winner this week. Take care, everybody. Bye.